Hi, Frank. How are you? I opened the room early to give Dr. Mavridou some time to because she's still new to Clubhouse, so to give her some time to find the room.
Alrighty. Okay. Maybe should I stay or come back later?
Yeah, it's up to you. You can stay. It's like it starts in like 7 minutes, so yeah.
For the April 9th event, I talked to Rich Sportsman. I think you know who is, and he is interested, and since you also agree, I'll just add him as a co-host.
Yep. Thank you for doing that. Yeah, that's perfect. And next week we'll have a very interesting presentation. Did you see the link? I think I put it in the. So
There are too many events happening.
on Thursday, this I just made today. He answered me and wanted to meet right away. He looked at our website and was impressed with the guest speakers. So he said yes. That's how he said. That's what he wrote in the email. So I was kind of happy about that. He's a really great physicist. So he. Hi, Dispoina. Thank you for coming. We are a little bit early, so we'll just give people time to come. Just open the room ahead of time, so you would have time to find it. Thank you. Oh yeah, so on Thursday, he is. I sent you the paper. He developed a new theory of entropy to solve problems in material science, and he was a huge paper that just came out. So he's coming on Thursday. So I think you will enjoy that too.
Wow, that's fantastic. I'll definitely read the paper very soon, and give some and have some ideas. So by the way, your channel group, can you assign like admin to me as well, or just like there isn't that function? I'm not familiar.
Yeah, I don't know how it works. I'll try to figure it out. I don't know.
Okay. I wanted bring Rich. I don't know if you are mutual
Yeah,
friends.
so I wanted to add him, but it doesn't work. So I think I'll just make a new one because it gives me very few options. Hi.
Okay.
Or you found the button to unmute.
I'm managed, yes. Can you hear me?
Yes, we can hear you. How are you
Okay,
doing?
great. I'm good. How are you?
Good. Yeah, thanks for coming. I'll introduce you in a few minutes, around
Sure.
4 to 5 minutes, and then the stage is yours. So people will still come in. So let's give people some time, and then thank you for that with the altmetric. So I have been adding them to all the YouTube. Like I added them to all the YouTube and podcast stuff.
Great.
Yeah, so I will keep doing that now. So that's
fantastic. Glad to hear it.
That's good. It will benefit, all the other guests.
Yeah, everybody, for sure.
Yeah, that's so good. And yeah, how was your week?
It's been okay. The usual marking scripts. Teaching is killing me, but yeah. Other than that, okay. I think normal. What about you?
Yeah. I have a project. I cannot share too much, but
Sure.
that has been in the company been dragging a long time. So I was assigned to now step in and help the people to get the project going. So today was the first bigger meeting, and it was a lot of conflict.
Okay. Sounds stressful.
Yeah, I just, because the boss said I should come in and make a experimental plan, but then, I just come in to something that's already their project for like a year, so it's not easy.
Yeah. It's not pleasant, is it?
No, but, I can't just say no. So
Mm-hmm.
anyhow, I mean, it's a fun project, but I understand that people get like,
Yeah, I think that's normal.
yeah. So, yeah, other than that, everything was good. The weather is finally good here. I don't know how it is.
Well, we are quite blessed.
Yeah, I
we
know.
have like 80, 90 degrees, so we can't complain really.
Oh, wow.
Yeah, well, it is Texas.
Yeah, it is Texas, so.
So yeah, it's now summer mode at this point.
Yeah, year was 2, and it's immediately everything is full because where I live, it's close. It's Williamsburg is relatively close to the new parks by the river they made.
Yeah.
So it's crazy to find the parking, so many people. Sometimes I enjoy when it's raining. There's nobody there.
Yeah. I'm liking the campus being empty for spring break, I can tell you.
Oh yeah, how is already?
Yeah, we have spring break now this week. It's finishing now. We're back in classes next week.
Yeah, already. For that.
Only 1 week.
Yeah, my kids will have spring break soon.
Yeah, we are not synchro with the rest of the country. That's true. So.
I don't know what to do with spring break and that weird second winter break.
Well, I know what to do with it. I can catch up on my to-do list.
Yeah, but with the kids,
Yeah, that's a problem.
they are not good camps this time. Like not like in the summer. And
Yeah, it's too short.
I used to just bring them into the university, and since especially my son, he like talks like a grown up, so he would have like real conversation with all the PhD students and everything.
I guess he entertained them, right? Like having him around.
Yes, exactly. So that was, but I cannot do that at the company. So.
Yeah.
Hi Eric, how are you? Sorry, I was pinging people and I wasn't checking.
Oh no, that's okay. Yeah, hi. Yeah, awesome title. Yeah, looking forward to it.
Thank you. Okay. Okay, we can slowly start and introduce, and then I think people will start coming in. What do you think?
Sure. Yeah. Absolutely.
Okay, so welcome everyone to the Science Society. We have another evening of really amazing guest speaker. We are very honored to have Dr. Mavridou. And let me introduce you and give you a little bit of background information. Dr. Mavridou is an assistant professor in molecular biosciences, and she is a chemist by degree, and she finished her undergrad studies at the University of Athens, and she then did her PhD in biochemistry at the University of Oxford, where she worked with Stuart Ferguson and Christina Redfield. And already back then, her project was about structure to function studies of Escherichia coli, DSBD, central protein for bond formation in the periplasm of gram-negative bacteria. And then after finishing her PhD, she remained for some time in the same research lab as a postdoc. And then she branched out to in 2014, she was awarded the MRC Career Development Award to start her own research group at the MRC Centre of Molecular Bacteriology and Infection at Imperial College London. And she also spent, before starting her fellowship, a year as a research fellow in the Department of Zoology in Oxford, as part of the research group of Kevin Foster and Stuart West. And there, at the Imperial College, Dr. Mavridou led her own research team and performed studies in the area of redox biology, bacterial antibiotic resistance, and polymicrobial community composition. And then she joined the Department of Molecular Biosciences at the University of Texas in Austin as an assistant professor in March 2020. So, yeah, thank you so much for coming and sharing your very important and very interesting research with us.
Thank you, Katarina. That's very nice of you and a very nice introduction. So hi, everyone. My name is Mavridou, and please feel free to interrupt me at any point. I like questions, and I'd prefer to chat with you rather than just talking myself. So I guess I should introduce what we do and what this work is about. So as most of you know, antibiotic resistance is a big problem. This problem is a major threat for modern medicine for two reasons. First of all, bacteria are becoming more and more resistant to the antibiotics we currently have. And at the same time, we have not really discovered many new drugs since, the 80s, 90s, really. We have a huge antibiotic discovery void that has been lasting for a long time. And currently, we need to find new antibiotics to treat resistant bacteria, but also we need to make sure that we find new strategies to protect the antibiotics we already have. And this work that was published recently is mostly about that. It's mostly about allowing the antibiotics we have to work better. So to back down a bit on that, a large number of bacteria, and in fact the most critical bacterial pathogens when it comes to resistance, are what is called gram-negative bacteria, and you will have heard some of them like E. coli, Pseudomonas, Klebsiella, Acinetobacter. Those are bacteria that are all gram-negative. Have one common characteristic, which is that they have two membranes. They have an outer membrane, a space, and then an inner membrane, and then the inside of the cell. So the fact that they have these two membranes makes them really difficult to treat with antibiotics. And in fact, these two membranes are a big barrier for antibiotic discovery. So along with the fact that these two membranes are protective, it's also a problem that in between the two membranes in this space that's called the periplasmic space, exist a lot of resistance proteins. So these proteins have the capacity to either degrade antibiotics that enter the cell or pump them back out or modify them so they don't work anymore. So they're definitely a big problem when it comes to resistance, because they are allowing the bacteria to incapacitate the antibiotics we're using. So a lot of efforts have been made recently to somehow deal with this problem of this particular bacteria, which are at the top of the WHO list for critical pathogens. And there's a number of ways people have tried that. One way is to try and attack the outer membrane by using compounds that permeabilize it, so that basically you only have to go through the one membrane, the inner membrane. Another way is to try and inhibit a lot of these resistance proteins that are sitting in between the two membranes. And there's a number of inhibitors that are used even clinically that actually bind to the resistance proteins, and then they incapacitate them so that then the antibiotic can work. And usually those are called adjuvants, and they are co-administered with antibiotics. And you might have been given augmenting. That's a very known combination of basically amoxicillin with clavulanic acid. The amoxicillin kills the bacteria, and the clavulanic acid inhibits resistant proteins so that the amoxicillin can work. So this is all well and good. Those are all good approaches, and at the moment we should try everything to deal with resistant bacteria. My lab has decided to go a different route, and that has been to instead of trying to either inhibit specific resistant proteins or trying to remove the outer membrane of bacteria, we wanted to see if we could prevent these resistance proteins from folding to start with. And we hoped that by doing that, we could actually target more than one resistance mechanism simultaneously. Because usually what happens with adjuvants is that they only attack a specific type of resistance, because they're specific to specific resistance proteins. So a way to do that is to basically affect protein folding. So generally proteins need to be produced, and then they need to fold in the right shape so that they can be functional. And if you can prevent this process from happening, then you will end up with a non-functional resistance protein, and then the bacteria will be sensitive to antibiotic treatment. So from my PhD work, I have worked for a long time on a process called disulfide bond formation. And these bonds are important because they allow proteins to fold more efficiently and to be more stable. So I knew that this process is important for other processes in the cell, in the bacterial cell, and nobody had ever looked at whether it's important for the activity of resistance proteins. So that's what we wanted to try. We wanted to see if we could inhibit disulfide bond formation, which is a process that allows proteins to become functional, whether we could actually kill too many resistant proteins at the same time. And this indeed worked. We inhibited the main protein that forms the sulfide bonds in E. coli, and we could see that a number of resistant proteins were affected, and they could no longer protect the bacteria from the antibiotics. So this protein is called DsbA, and it's just a small protein that handles a lot of other proteins and helps them acquire the sulfide bonds. From what we first did is we deleted it, and then we looked at how the resistance proteins behaved, and we found that a lot of them were degraded. So that meant that they were not around anymore to protect bacteria from antibiotics. So that was the first observation we had. And then we basically took these results into a more clinically relevant setting, which was to test the exact same thing on strains found from the clinic, so on clinical strains isolated from patients that were naturally resistant. And we found that even there, when we could inhibit or delete DsbA, we could actually stop the strains from being resistant, and we could treat the bacteria with existing antibiotics, and usually antibiotics of last resort. So antibiotics that you would get if you were in the ICU, if you had a very serious infection, and that are particularly precious at the moment because they're the only thing we've got for very resistant bacteria. And the last thing we did is we tested the same thing in an infection model. We didn't use an animal infection model. We used an insect infection model to start with, and we found that again, if you impaired the sulfide bond formation and you co-administered an antibiotic, you could clear multi-drug resistant Pseudomonas aeruginosa from insect larvae, and you could allow them to survive, whereas any other kind of single treatment, only antibiotic or only inhibitor of the disulfide bond formation, or no treatment at all, ended up in death of the insect larva. So this is a preliminary evidence that this could potentially be a useful therapeutic avenue in the future.
Question.
Yeah.
So yeah, I mean, it's a fascinating approach. I'm curious though, how you incorporated selectivity in your strategy. There's a lot of proteins with disulfide bonds, and imagine, in terms of a therapy, if it, can't go around ripping open every disulfide bond. Curious, what was selective about the protein where you open the disulfides?
Sure, very, very good question. So we basically DsbA is bacterial specific. So while disulfide bonds are indeed needed for basically many proteins in humans and in animals, by inhibiting DsbA, you don't really affect any of these proteins because they are formed by different kind of pathways that would not be inhibited by a DsbA inhibiting approach. So in this case, we are only affecting disulfide bond formation in bacteria. And in this case, that basically leads to the degradation of a number of proteins that are in the envelope of the bacteria. So they are proteins that are mostly localized in between the two membranes. So if you normally inhibit DsbA in bacteria without antibiotic treatment, the bacteria don't die. DsbA is not an essential protein, not even for bacteria. But when you inhibit DsbA in the presence of antibiotic, because you have basically not allowed the resistance proteins to fold, you then kill the bacteria through the action of the antibiotic. Is that clear?
But was there something specific that got them into the bacteria in the first place?
No. So the DsbA inhibitor will go everywhere, but it will not inhibit anything on the human body because there's no DsbA in humans.
Okay, so it was specific for the DsbA. Yeah.
Yes. So either we deleted DsbA from bacteria to do experiments in the lab, or when we administered an inhibitor, is a DsbA inhibitor. So the only target you inhibit is that one protein in bacteria.
Cool.
Thank you. Any more questions?
It's yeah. If there aren't any more questions, let me just ask another.
Sure.
Were you looking at linking them to, well, I guess, is the site of action anywhere near the membrane, and would it make sense to link the or for more targeting, link the amoxicillin or whatever the the antibiotic proper is?
To the inhibitor?
Mm-hmm.
Yes, absolutely. These are approaches we're looking into. So basically, DsbA is sitting also in the same space. So it's sitting in between the two membranes. So actually, whatever inhibitor you would need to use for it would only need to go through the outer membrane. And yes, trying to link antibiotics to inhibitors for DsbA could be a way forward, especially because amoxicillin has a way to find bacteria because it usually goes through routes like the urinary tract, where you often have, for example, infections, and it's a good way to target specifically pathogens. Yeah. So are there any particular? Yes.
Yes.
Tell me.
Thanks. I have a question follow up from Serena.
Mm-hmm.
If it's the DsbA is it just like specific to this particular bacteria that you study, or it's or it's also it's a family of bacteria that has DsbA?
Yeah. So DsbA is quite conserved in many bacterial species, very conserved in gram negatives, which are like the most critical kind of pathogens we have at the moment and we need to deal with. But it's also found in gram positive species, which is more like Staph aureus and strep species. So it's quite conserved. It could, target could lead to treating multiple bacterial species. I'll be honest here, and I would say it's not you could not deal with one inhibitor for every species, because even within bacteria it changes and it's diverse within the bacteria phylogeny. So you'd probably need multiple DsbA inhibitors to be able to deal with any kind of infection. So either you need a mix of DsbA inhibitors, or you'd need to know what pathogen you're dealing with and administer the right inhibitor. So I don't think you could be able to have a broad approach with a single molecule that would inhibit every single bacterial species. But you could do with broad classes of inhibitors.
I see. And then if that in that case, is your algorithm or methods can be generalized to similar DsbA, quote unquote, in other, broadening, the application of your approach. Maybe, other bacteria has only single wall, right? So just curious.
So with bacteria that have single walls, DsbA actually sits on the surface. So it's anchored to their membrane, their single membrane, and it sits there and it processes. And a lot of the resistance proteins also actually sit in the same space, so they are anchored to the membrane. So again, it would be the same case. Actually, the DsbA would be pretty exposed, and it would be easier to inhibit that one. But I don't think that the same inhibitor that would inhibit an E. coli DsbA would do the same for a gram-positive species like a Staphylococcus. I think you can group bacteria in categories, and you could probably deal with a number of important pathogens with one inhibitor, but then you'd need a different inhibitor for another class of pathogens, depending on how similar their proteins were.
Thank you.
For the case where it's membrane bound and exposed, I suppose one strategy would be to put a lipid tail on it of appropriate composition for those membranes.
I don't think you need to put anything on the inhibitor. You wouldn't need really to put anything. I think the DsbA would be accessible to the inhibitor immediately. So I think it would be even easier to inhibit it in those cases, actually.
And was this a success story with computer-aided molecular design, or did you just discover these in some other way?
Not at all. I mean, it was a completely hypothesis-driven approach. So we did not use any computer methods for this. And it was based on the fact that we understood the biology of the bacterial species. We knew that this particular pathway was important for a number of housekeeping processes in bacteria. We knew that the same pathway was folding proteins involved in pathogenesis, like formation of toxins, formation of adhesins, and things that bacteria use to cause infection in the first place. And it's just that nobody had ever looked at whether the same pathway is important for the formation of resistance proteins, even though the resistance proteins are localized in the same environment as all these other proteins I'm talking about. So it basically was staring us in the eye. It's just that nobody had ever tried it.
So that's awesome. That's a great example of like analytical approaches to science still being viable, that it's not all just computational approximations. That's really exciting.
Of course. I mean, and I have no issue with AI and computational approaches. They're very useful. But I think you will still go back and you will still need to understand the physiology of whatever organism you're looking at. And everything is going to go back to whatever you discover, even if in examples where they have discovered new compounds using AI, and the compounds indeed are effective and indeed are killing bacteria, for example, and they could be potentially new antibiotics. You can do nothing until you understand their mode of action. Nobody's going to go forward with compounds that you don't understand how they work exactly in the cell, and that will bring you down again to physiology and an understanding of biochemistry and molecular biology. So we will never escape from that.
So this pathway is invariant with respect to the change or mutation that is instigated by the antibiotic resistant changes.
Can you say again, because I missed you in the beginning?
Okay, so is this pathway invariant with respect to all the changes undergoing in in the bacteria with respect to the antibiotic resistance?
So you're asking whether we could develop resistance there, right?
Yeah. So is this like is this intrinsic to the survival of the? Well, it cannot
So
possibly change in with respect to the survival of the bacteria, and will not change even if it develops various antibiotic resistance.
So that's the problem. Like, you never know where the resistance will come from. And it's not unlike, we don't have a good enough inhibitor now to be able to do the studies to see if we can get resistance arising. The good thing about this protein as a target is that it's not essential for bacterial survival if you don't have antibiotic present. So in a way, the selective pressure is smaller on occasion where you don't have antibiotic treatment, which is a problem that we normally have with antibiotics, that basically you are exposing all sorts of bacteria all the time, from clinical waste, from environmental waste to antibiotics, and then you get mutations enriched even when you're not actually actively doing any treatment. So with a DsbA inhibition, you will not be applying any selective pressure to bacteria unless you have a relevant antibiotic around. But whether you will get resistance to an inhibitor of DsbA, I can't say unless I have an inhibitor that's clinically applicable, because resistance can come from many avenues. So it could be from things that you don't expect. The fact that it has many functions as a protein is promising, because usually the bacteria don't know how to deal with something like that. But that doesn't mean much. So like, until we have an inhibitor and we know exactly what that could be, we can't say. People have found some DsbA inhibitors for other reasons, and for those ones, when they have tested them, they haven't found resistance. But they haven't done tests under antibiotic exposure, so it's hard to say now.
I see. Thank you.
What were the efficacies you found? Is this micromolar or better?
So we did not use, so either we deleted the protein in the gene for the protein, in which case, that doesn't apply, or we used a laboratory inhibitor that we would not be able to use in humans. And there, the amount that we used to have inhibition was 50 micromolar.
Okay.
It's a good inhibitor, but it's just not appropriate for clinical use.
Mm-hmm.
It's a good tool for the lab.
And can you comment on the chemical class? Is it small molecule, or
Of the inhibitor, yeah, it's a small molecule. Actually, it doesn't inhibit directly DsbA. It inhibits its partner protein. That's why I'm saying it's not good for clinical use. So DsbA, the way it works is it makes a disulfide bond, and then it needs to be regenerated from another protein called DsBB. And then that molecule inhibits DsBB. So there are a number of problems with it if you were to use it in tissue, for example, in a human or even in an insect. So it's not applicable for clinical use. It's a very good tool to use in the lab for when you can have completely defined conditions of bacterial growth.
Well, so it may serve as a lead compound for a more thorough computational study, for example.
Not really, because DsBB is completely different to DsbA. But there's other DsbA inhibitors discovered through other approaches. There's some small molecule ones, and there's a peptidomimetic one, and those could serve as lead compounds. The other problem until now for DsbA inhibitor discovery has been that it's much easier to discover inhibitors if you have conditions that kill the bacteria to screen for. And until now, we didn't have them. Like I said, DsbA is not essential. Now that we know that if you treat with the right antibiotics, you can get bacteria to die if you inhibit DsbA, it's much more easy to screen broadly. Until now, that was not easy, and they were screening at the level of the protein, which then leads to compounds that don't necessarily get into the cell. Does that make sense?
Yes. And I'm wondering, from your description, this sounds like Yeah, you mentioned it was, staring at us all along. I, first became aware of the, problem in the 90s when I was working on my chemistry doctorate, ironically in computer-aided molecular design. But I was struck by how much, how little interest there was from the pharmaceutical research departments in pursuing this. We would regularly hear seminars about how much of a problem it is, and often with doomsday tones. But and there would be cynical commentary from the presenters that, in essence, the lack of interest was business model related. They wanted to wait until it was a problem to solve it,
It
and
still is.
it still
Still the
is.
same reasons. It's exactly the same reasons that drive this. And now there's no pharmaceutical companies that are in that they actually officially have mostly abandoned antibiotic discovery. Genentech is one of the few companies that is still doing it because they think they should. So more as a matter of principle than anything. And basically we are relying on academia and small biotechs, and it's not looking good because the problem is bigger. We have exactly as many antibiotics as we had 10 or 20 or 30 years ago, and all the big pharma is out officially after Achaogen closed. That was their exit point. Yeah, and it's because it's very difficult. So they know that there is basically you make a new drug and you lose money, and it's anyway very difficult to find one. And then when you market it, you lose money. So and then it doesn't last long because resistance arises. So without incentives, without push incentives from federal governments, it's very difficult to convince big pharma to invest money in this.
So how long was that journey for you from when you first kind of either stumbled upon or had the realization, the eureka moment, to when you finally saw it work in the lab?
Well, I mean, from the moment we started the work, I guess it was in 2017. We published it this year, so it's about 4.5 years. But you have to keep in mind that I was a very young group leader. I had a group of 2 people. We started with nothing, basically two E. coli strains and no tools whatsoever, no experience with antibiotics. I was a basic biochemist before that was working on kind of the pathway rather than anything um, to do with the bacterial physiology. Yeah, and since then, I've moved continents. COVID has happened, so yeah, it could have been done maybe 3.5 years. So more or less about that, which is not atypical for most of these kind of things when they are from
Yeah,
scratch.
no, that's impressive. Usually, I'm familiar with like it taking a decade to go through that process, so it's exciting to see it accelerate, even though you don't necessarily use computational tools. So I think one of the projections is usually computational methods have the time. So a PhD taking 4 or 5 years or that kind of body of work can be boiled down to like 18 months to 2 years. I think that's a little ambitious, but this is very exciting. I did have a question about like something like bacteria that have other mechanisms, because you mentioned this was one of the mechanisms. Could you briefly comment on other mechanisms of antibiotic resistance?
Sure. So this particular approach is applicable to only a specific set of resistant proteins. So it mainly affects beta-lactamases, and so beta-lactamases degrade beta-lactams. So beta-lactams are drugs like penicillin and all their derivatives. So it will be effective against many beta-lactamases, not all. Only the ones that have disulfide bonds, but it happens that a lot of the very clinically relevant ones do have disulfide bonds and are dependent on the system. And then colistin resistance, which is a completely different mechanism of resistance, and it has to do with a last line treatment with a drug colistin. And this currently is a drug that is under threat because there's a new mechanism of resistance that is generally easily disseminated. through mobile elements. So bacteria can exchange this mechanism of resistance, and we have seen increase in resistance. And this is a very, very last resort drug. So we can actually deal, that particular approach can actually incapacitate these enzymes as well, all of these enzymes and any new ones that might arise, because they all have multiple disulfide bonds. So beyond those, I mean, we looked into efflux pumps and we see a small impairment there as well, but that's rather unimpressive compared to the other two mechanisms that we can really affect. And that's about it, I would say. But that's enough. So what I would say is that if you could salvage lactams, and you're already in a good shape, because that's all we need. Lactam antibiotics are all we need. Like, they're really good drugs. They work against everything, and they're under extreme threat because we use them all the time, and we have like 20 or 30% resistance against them. So just salvaging those is sufficient. And then colistin is a last resort drug that you'd use if you could not use lactams. So that already is good as well.
So there is like an alternative approach with phages. I guess that's usually a difference in between East and West, kind of. They tend to sometimes go in different directions. Do you see your approach becoming more mainstream and effectively replacing something like the previous standard or previous protocols? I know it's a little early because you said it was smaller models, not yet human models, but what do you
Sure,
see as
we
the
don't have an
potential
inhibitor
timeline?
yet. So we are nowhere near, we are looking for, like, we are planning to look for one, but we don't have an inhibitor molecule. So for now, this is just a strategy, a proof of principle, a potential way forward. But without a molecule, you can't really say that this will replace anything that is already in use. When you said phages, though, you mean use of phage therapy?
Yeah, so a phage can either disrupt the metabolism of the bacteria or cause some other critical error.
Sure, a phage will kill a bacterium, but there's very limited proof of use of phage therapy successfully, except topically, potentially. So the majority of ways we have to deal with bacterial infections is through the existing antibiotics we have at the moment and adjuvant antibiotic combinations. That's the main paths we have forward for dealing with bacterial infections. Sure.
Did you still have like did you like already summarize all of what you wanted to talk about? Should we open it up to more questions, or do you want to continue?
Sure. I'm happy to have more questions. I mean, there's nothing more to it. This is all that there is. I mean, I can go into more details about specific examples if people are interested in specific things. I'm happy to discuss, but broadly, this is what the paper has showed, that basically that you target a single protein and you can incapacitate multiple other resistance proteins, and that allows us to treat multiple pathogens for multiple mechanisms of resistance.
Yeah, I see that Dr. Shah and Katie and Janelle, like we have various people here on stage that didn't get the chance to ask a question, and Frank, so please go ahead and ask your questions if you would like.
I'm just here to listen, but thank you.
Okay, Dr. Shah, Katie, do you have?
Thank you so much, Katrina. I'm somehow in a crowded area. I'm just changing my spot, then I'll be back to ask my question. Thank you so much.
Of course, sure. Katie or Hansen, do you have a question?
I guess I'll ask one. From unfamiliar with as a layperson to this field, could you go in shed more lights on the procedures that I do hear that you mentioned you are not doing computational in terms of finding the molecules, but what is the there is some kind of you need to produce in the lab in the E. coli the with gene
Mhm.
expressions. Yeah, could you
So
give
how
some
did we
detail?
investigate that in the lab?
Yes.
So basically we started from the basics. We first wanted to use laboratory E. coli that we knew was really not resistant, and we had like it was completely devoid of resistance mechanisms. And so we used that E. coli, and then we constructed it so it was producing the resistance proteins that we were interested in. And we did that by using the genes from the particular resistance proteins that we either synthesized or we got from existing clinical strains. And then we cloned them into a vector of choice, which is just a circular piece of DNA that will actually allow the protein to express in the laboratory E. coli. And so in this system, you have a very naive, susceptible strain that will then become resistant because of the use because of the presence of this gene that makes that protein. And in fact, we could see that you can measure how resistant a strain is through exposing it to antibiotics and looking at what level of antibiotic concentration you can kill it. So you use this assay routinely to see how resistant a strain is. And after we introduced these genes to these strains, we could see that they became more resistant. And then we constructed strains that were lacking the target protein, which is DsbA, so they couldn't make the sulfide bonds anymore. And then we could see that as soon as we did that, the resistance of these laboratory strains went back down. So we knew that the likely that the resistance mechanism were affected. After we did that, and we did a number of control experiments to make sure that this was specific to these particular resistance proteins, and we were not like ruining the cells or we're not affecting their metabolism. We did all sorts of control assays to make sure that we were not affecting their cell physiology beyond the assembly of this resistance protein. Then we looked at the resistance protein specifically. So you can add specific small tags at the end of the protein that you can then that will then allow you to visualize it. So we could actually see that in the strains that could make the sulfide bonds, the resistance proteins were present, and in the cell.
Some fluorescence.
No, just affinity tags, so much smaller than fluorophores. So you use a standard immunoblot against antibodies, so they could be as big as 6 or 10 amino acids. And then you just perform electrophoresis of the total protein content, and you just see whether you have your protein there or not. And what we saw is that if you could form the sulfide bonds, the protein was there. If you could not form the sulfide bonds, the protein was not there anymore, or the amount was really vastly reduced. So that's where we were with the, let's say, completely laboratory side of the project. As soon as we had that and we were certain, and we tested that against about I think 12 or 11 lactamases and 5 I think colistin resistance proteins, so we knew that it was a broad effect across many resistance proteins. Then we went into looking at whether we could recapitulate that using the inhibitor molecule. So we purchased the published inhibitor for the partner protein. We first tested that it was working in our hands. So DsbA also affects bacterial motility. So that's the first thing we tried. We tried to see if the bacteria were becoming non-motile. And as soon as we could see that that was the case, it meant that the inhibitor was working in our hands. And then we applied that to the same strains we had used before, so the laboratory strains, that just to see that the same results that we got with the deletion of DsbA would get with the inhibition of DsbA. We saw that that was the case. And then we started applying them to clinical strains. So we had a number of clinical isolates. We knew what resistance proteins we had in there, and they were different species. So we applied the inhibitor to these species, and we saw that basically we're getting the same, or actually larger effects there. We could see that basically there we could actually assess whether you could go from a sensitive from a resistant to a sensitive strain. Since there's published guidelines about the level of resistance that would allow you to treat or not with an antibiotic, then you could go and check exactly whether you were going from a strain you could treat to a strain you could not treat to a strain you could treat. So we had big drops in their MICs, as they called, which are the minimum inhibitory concentration for drugs. when we were applying the inhibitor as well. Is that clear?
Oh yeah, thanks. It's very fascinating. It's very interesting. So just in terms of what you shared, the uh, uh, just try to see if I got it right. So the
Mm-hmm.
so for your algorithm, your the approach that there are probably other and you know resistance antibiotic resistance mechanism that are outside. So yeah, at this point, you can clearly draw the boundary of the scope, and
Mm-hmm.
you can enumerate. You know what type of proteins that are dependent on the DS. I mean, in your approach, you can just enumerate what will be effective, right? So
Yeah.
any other important um target lying outside, maybe your next or or anything that
So
worthy.
So I think we've covered. So we have a number of proteins covered in this first publication. Um, there's a next one coming soon that has another bunch of them. It's the same types of proteins, like meaning it's the same mechanisms of resistance, just different. So like with beta-lactamases, those enzymes that break down beta-lactam antibiotics, there's 6,500 enzymes currently identified to date. So it's a huge space. You can classify these enzymes into about 500 phylogenetically different families, so discrete families of enzymes. And we can affect quite a lot of these families already from the first study, and we are looking at some more, and we are looking at some that are more infection specific, so that we might apply more for, say, for example, cystic fibrosis patients. It's the same mechanism of resistance, just different enzymes that apply in different scenarios. So we're moving towards there to explore the whole space. With colistin resistance, we have covered all of the span of these proteins, and we are pretty certain that all of them are affected if DsbA is not there. So we can predict that even if new ones arise, and they do arise regularly, those will be also affected. There's not many more mechanisms or resistance in that space that DsbA would affect. But there's other similar proteins like DsbA that do different kind of processes that allow for proteins to fold correctly that could be targets for other kind of resistance mechanisms. And that's something that we are looking into going into like in the future, but it's a whole different project. We're still working. We're still working on this particular aspect. We still have stuff to do here.
Well, thank you.
So you mentioned in the paper that I think there's one anti-rheumatic drug
Mm-hmm.
that maybe
Or
could,
nothing. Yeah.
yeah, but that's the only one currently known, right? Is there anyone trying um to use it?
For MCRs, yeah. So ranofin was found through a screen of FDA approved compounds as a existing approved drug that can actually impair MCR and proteins. So one of the two enzyme mechanisms we're talking about, and a few beta-lactamases, only one class of them. That paper got published in, if I remember correctly, in 2021, and I haven't heard whether it's not in clinical use for resistance yet. Obviously, it's an approved drug for rheumatism, but it's not something that to be repurposed they will have to go through a certain level of trialing, I think.
Yeah, I hope
But
that,
that was also very promising. Like it had very promising specs as well. The results were very exciting for that paper.
yeah, that's interesting. Maybe they could start trying it in very cases that are quite hopeless, by approved just by a single patient and start this way.
Compassionate use.
Yeah,
Exactly. Yeah. I'll tell my
that's how phage therapy has been used in the past in a few successful cases, and it was successful, although it's not clear exactly what happened, but yeah, it was successful in principle.
Okay. Yeah, it's not very helpful, for doing a systematic study, but just to get
No.
started and get people interested in it. Sometimes those case reports, if they get attention.
That's useful because it's an FDA approved drug, so one could actually do that. Whereas with something like our inhibitor, like it's not even our inhibitor, something like the inhibitor we used is not possible because that's not approved in any way or effective in any way in mammals. So yeah, it's much more difficult to try that there.
I had a question.
Sure.
So you had mentioned that there are 6,500 enzymes identified to date with about 500 classes. So the implications you're finding apply to how many of these?
I can pull up the statistics we have. So we are comparable to other existing adjuvant approaches. Let me pull up my notes so I can tell you exactly without. Let me find it a second. So, give me a second.
We're pleased to wait for good information.
Sure. Table. We had a table with a comparison compared to other adjuvants. Yeah. So basically, classical inhibitors that have been in the clinic since the 80s and 90s can inhibit about 125 families. New generation inhibitors have similar success rate, about 190 families of enzymes. The difference is they can do more enzymes, like they can do more different enzymes sometimes.
At the same time?
Yes.
Got it.
And then in development, there's a particular inhibitor called, for example, taniborbactam that is phase 3 clinical trials, and it's very exciting because it can basically almost do so β-lactamases can be divided into 4 empirical classes, A, B, C, and D. And B is a particularly difficult class that is further divided into 3 subclasses. So taniborbactam, which is in development at the moment, can do A, C, D, and B3. B2. And that's about 276 families. So our approach would do about 180 families. The difference is that it can also do a few families that are nothing else can do so far from the big clade. And again, one thing to keep in mind is that we're not competing with these other approaches. They're all needed. so this one that is in development and it's hopeful is very exciting, but often even if these adjuvants are put in clinical trial and then are put into practice, you get resistance against even the adjuvant, so both the antibiotic and the adjuvant. And so the more you have, the better. It's one of these cases where they keep being developed, especially against these enzymes, because they're extremely precious. So yeah, it's not a competition. So we are on a similar level. But we have some enzymes that other approaches don't have, which is good because that means that you have a broader spectrum of inhibitors that you can use.
Cooperation over competition sounds like evolution to me.
Depends what you mean. Bacterial competition is more prevalent, but hopefully in humans that's not the case.
I meant sort of at the macro scale of things.
Yeah, the macro scale, yeah, that's true. I mean, this is one of these cases where the more the better. Like it's not useless to have more options with these infections.
Absolutely. Thank you.
How's the funding situation for this kind of research? Is there a lot, or is there still not enough attention to this problem? Like, does the Bill Gates Foundation or so give money, or like they did with the COVID vaccine, or is it still not in the focus of people?
I don't think the Bill Gates Foundation gives much on resistance, actually, to be honest with you. So personally, I'm funded by the NIH at the moment, but still the research we do is on the fundamental level. The problem really will be when, if potentially we had a molecule that looked good and it was a potentially promising inhibitor. For example, it worked on bacteria and it killed resistant bacteria along with an antibiotic. We happened to show that it worked in mice and it had good cytotoxicity specs. There we would be in a problem because that is the space where there's the gap of funding. So while you're still trying to develop something at a fundamental level, you can get funding. If you're past the preclinical trial level, you can potentially sell or give your invention to something like a pharma company to develop it if they were interested. But in between, that's where a lot of things die off. So we, as a lab, we're not in this position yet because we don't have a potential molecule. So we're still funded at a fundamental level from the NIH.
Yeah, that's not the first time I hear it. A colleague and friend of mine from Italy, she's a biochemist. She develops a lot of epigenetic drugs.
Mm-hmm.
Are really great. I mean, there's one she developed for chronic pain, for severe chronic pain. It looks great in mice. Like you
Mm-hmm.
don't have the addiction issues and
Mm-hmm.
drowsiness and toxicity looks great. And she doesn't get funding to continue.
Yeah,
This
that
stuff.
space is a huge problem, and that's why these pull and push incentives are implemented. For example, there's Carbex now, which hope another organizations like Carbex that hopefully they do actually provide much more funding for smaller biotechs or even research groups in universities to push their discoveries through to the next level. I think that's working. The push incentives. The problem we will have again with that is that when those even get pushed further down the line, a big pharma has to take that on for producing it and marketing it, and they will still lose money. So even if we had a perfect drug, I doubt that it would be that easy to put on the market. But I guess we're not there yet. So
Yeah,
How much of
one
drug
place.
development goes to, how much of a drug development budget goes to marketing? I think it's substantial.
yeah, I meant more like it's proven now that if you produce an antibiotic and put it on the market, you lose money. It just happens. That's what happened to Achaogen. The company had a huge amount of value while they were developing their drug, and as soon as it hit the market officially, their stocks plummeted and they closed down. Because the antibiotics are cheap and there's no incentives to prescribe more expensive ones if the older ones work or could work, and there's a push from insurance companies to keep doing that, to keep giving the cheaper ones. The older ones are cheaper, and so you can imagine if you basically as a pharma company, you take something that was in preclinical or phase 1 clinical trials. I think that's the best a researcher can do, bring it to that level, and then takes it to do phase 2 clinical trials, phase 3 clinical trials, and put it on the market, produce it and put it on the market. This is still millions to billions of dollars. At that point, if then it's not sold, or if it's not sold expensively enough, then they will not make money. They will lose money, and so that's why they don't want them anymore. So they need to be subsidized by other sources, governments usually.
I guess what I'm saying is that I feel that science should be funded properly, and that's clearly not the case at this time.
Mm-hmm. I think still, I find that for research around resistance, there's a general acceptance from the funding bodies that this is a big problem, and there is support. But with antibiotics, the problem comes more down the line, I think, than the initial discovery. And that problem is known to exist, and it has to do with the fact that it's very hard to convince companies to lose money for producing products. They need to be incentivized otherwise.
Yeah, I know of one area where there would be significant demand for this. In fact, I back channel messaged you. It's a talk by a DARPA program manager, and he talks about how the warfighter, the soldiers who are at the front lines, when they get injured, not like their injuries sometimes make them succumb to infection, and through the treatment of antibiotics so early on, the ones who do survive that process end up having this critical infection anyway. So they went through all of this sacrifice and suffering, and then in the end, it's a little microbe that gets them. So I think that's an immediate application for that, and there's probably a lot of incentive there. So you should
Sure.
definitely try to connect with that program manager.
Yeah, DARPA is on my radar
Or you're on
for
theirs.
funding this. Or I'm on theirs. Well, I hope so. But yeah, the soldiers have specific problems, especially with Acinetobacter, which is a very nasty bug, which commonly is found in sites of battle sites and infects them, and they're incredibly resistant. So yeah, I agree.
What about the meat industry? I mean, they use antibiotics all the time, but probably they want to use the cheapest one, and probably
Yeah, they do.
there's a lot of resistance there.
They actually contribute to the problem.
Yeah, that I know.
So yeah, absolutely. So colistin resistance, those particular proteins that we study that are mobilizable and they move around, the genes move around, they came from the agricultural industry. Colistin didn't used to be used in the clinic because it's very nephrotoxic, so it causes kidney toxicity, and basically that's why it was used extensively in agriculture. And in 2015, these new resistant proteins were identified that were jumping around in multiple strains in the clinic, and then 2 years later, colistin re-entered the clinic because we need it, because sometimes there's nothing else we can treat with. And now we have this problem that basically came from agriculture. Now they have stopped using it in the animal feed, but the mechanism is now there.
Yeah, so maybe the governments have to force them to use an inhibitor or a resistant breaking compound, and then that would be a way to make money. No, I don't know.
Yeah, that would probably be like a
Potentially.
regulatory framework. That's a really good as a countermeasure.
Maybe they should make them pay for the damage that they've caused.
Or that, yeah, like the nicotine, like the cigarette companies and so on. That's a good idea.
For now, it's good that they've stopped doing it because, within.
Well, what are they
6
using
years,
instead?
we
They
have
haven't just stopped responding to the threat. The threat hasn't abated yet.
Well, they stopped using that particular antibiotic, which is extremely precious in the clinic, and that will help with the resistance kind of jumping around everywhere. To be fair, like, this resistance mechanism came out of nowhere, and we still really don't know where it's coming from, why it came like that. So it wasn't really, there was nothing nefarious here. I think that it was bacteria can always get around whatever you throw at them to kill them. They're ahead of us.
I bet if you were to dig into their internal documentation, they were certainly aware of this problem.
It's possible.
I bet.
Yeah, I
Probably a
can't
good bet.
comment. Sorry?
I just said it's probably a good bet, but a little bit conspiratorial, because I don't think some companies may accidentally have stumbled upon this. So I want to make sure we're not painting all companies with the same kind of tinfoil hat there.
Well, I mean, I'm just overgeneralizing based on my experience with documents in court, based on various different industries being aware of the threats that they're posing. But we shall not get too tinfoil hat.
It certainly took a while to stop this practice. I will agree to that. And the resistance was there already, and we knew about it for a while. But also, things are very slow to change in all of these contexts. So until something is clear to be a very important threat and really a big problem. It takes a while.
And it has to be demonstrated in court, usually beyond a reasonable doubt, kind of like how tobacco took a while. And just the joke about tobacco scientists is that they could disprove gravity. So sometimes you're working against a very strong lobby.
That's a great example. I mean, tobacco is a perfect example of internal documentation proving that they were aware of the threat. But we digress.
Yeah, I wanted to check in with you because we reached like an hour, so that's I think the time you have available. So I don't know if anyone has a last question or so, please
I have one question.
go ahead.
So, eventually, so my question. Thank you so much, first of all, for sharing the fascinating topic. And my question is, most of the approaches happen about the proplasmic, actually protein, right?
As far as we go, yes.
Uh-oh, connectivity issues.
Yeah, I think we lost her.
Yeah, I think so. I can write Dr. Shah or Dr. Shah, maybe you want to write us the question and then either I'll forward it or through email or direct messaging. I guess.
And I have time. I'm happy to stay for a couple more minutes. It's fine by me. If there are more questions, I'm happy to answer them.
Yeah, go ahead. Go ahead.
Yeah, so that's great. So let me ask one. I'm curious. Could you explain more? I mean, when you say share the feeling that the solution is staring at us all, but you're the somehow you there's a light bulb, eureka moment, as quoting Io. The uh, so as I understand so far is there is a particular family, one in the you call lactamase. I mean,
Mm-hmm.
so the traditional people who are not, previously they just think to come up with ever newer, antibiotics, and didn't
Mm-hmm.
think of. I mean, well, it's definitely, from the logic, if you actually destruct the underlying mechanism together, it's like you take off the headquarters, right? So then
Yeah.
seems to be definitely it's the staring, right? So I'm just shocked that it's a there has to be some difficulties or some conceptual or built in,
Well,
made us
I think
oblivion.
some. Yeah. So why had they not realized it? Well, I mean, this is in no way a criticism to the current efforts to inhibit beta-lactamases with inhibitors. I mean, they have been successful, and those are compounds that are in clinical use, and they are extremely important. It's just that they're more limited in terms of what they can target, how many things they can target, and sometimes you get resistance to them arising quite readily. But it's what has saved beta lactams from the 80s onwards, so it's been a huge effort. I think you need to have a bit of a niche expertise to realize what we realized. Like, I've worked on the DsbA system, which is the broader family of DsbA belongs to, since 2004. I know the system really well. It's been already proposed as a broad acting strategy to stop virulence, which is nothing to do with resistance, has to do with the ability of a bacterium to invade a host. And so for me, it was a small step towards the right direction. I think because DsbA is not an essential protein for cell viability and is a big gear towards looking for silver bullets and things that will kill bacteria off the bat, that's probably why that has never been on the radar. I have to say that I've once in a while I have bumped upon people who were doing kind of non-specific screens against resistance, and after hearing my talk, they would tell me, "Oh, now I understand why we were getting DsbA as a good target from our screens, but we never understood the physiology, why it was coming up as such a good hit for resistance." And like, again, you need to have the right background to appreciate um, that connection, and it's not a very, it's a bit of a niche expertise. I think that's why.
I see. Thank you. So you mentioned, I hear you mentioned the keyword heat resistance. Could you
Sorry.
explain? Did you say heat resistance, or
No, they were just hits. They would do broad screens using transposon libraries and try and find genes that would be affected. That if you affected these genes, you would get resistance phenotypes. You would get like lack of resistance, and DsbA was always coming high up on their screens, but they just were disregarding it because they couldn't understand why that was relevant. Because if you look at what it does, if you look at DsbA, if you Google DsbA, it will come up as disulfide bond formation protein, which doesn't give you any idea about resistance. So
I
they were
see.
just disregarding it, I think, sometimes when they were actually identifying it as a potential promising avenue.
I see. That's great.
Could you say the disulfide bonding what? DsbA?
That's what DsbA does. Yes, that's what it does. It makes disulfide bonds in proteins, and so it makes a disulfide bond in resistance proteins. And then if you don't make that bond, the resistant proteins crumble and they don't form at all. Katrina, there was has Dr. Sam managed to send her comment over?
Yeah, she wrote me that if you could share more about the proteomic approaches that you used. That was what she.
Meaning how we figured out that the resistance proteins were degraded is that we didn't really use broad proteomics approaches. We just looked specifically at the proteins of interest. So we looked specifically at the resistance proteins we were expressing, and we wanted to see if those proteins were stable or not in the absence of DsbA. So we used just immunoblotting against those specific proteins. So there was no proteomics in this, not any broad proteomics approaches really involved in this study. Does that answer the question?
Yeah, I think so. Yeah, I think so. Yeah, I'd like this. I know it's like fancy nowadays to write, used AI or some machine learning, but I think if we if we have like, I think still our mind, if we are like, well, like if we worked in this for a while, I think our mind is still better at finding good targets than random machine learning approaches, because it could just be random correlation that just happens for no reason. Also, I'm very critical sometimes.
I agree with you. I mean, there's two schools when it comes to antibiotic discovery. You have like completely cell-based assays where you throw at them compound libraries and you see what inhibits their growth, and you can use machine learning or not. But even if you're just doing it all empirically, it's just non-hypothesis driven approaches where you just take things through compound libraries and just see what inhibits cell growth. And then there's hypothesis driven approaches where you think that something should be a good target and then you specifically test it and then you hope to find an inhibitor. I would say both have been unsuccessful or both have been equally successful. You can call it either way.
Well, not
We
with
still
the
don't have
first,
drugs.
not with the first good antibiotic, right? Because history of antibiotics, when the first one was discovered, Germany, I think hundreds of scientists under Bayer and other chemical companies in Germany back then were just brute force trying all compounds out they had from actually coloring, right? Wasn't
And they
it?
discovered some, indeed. They discovered the sulfa drugs, they discovered natural products like all the naturally like originating antibiotics. But these spaces are now quite done. Like,
But was brute force, but the really effective one was not discovered by brute force. It was,
no, then was optimized, of
yeah,
course.
exactly.
Yes, of course. You always need to do that. Like that is we are aware of that. That is it needs thinking and optimizing. But now we don't even have that. We don't even have the the initial brute force directing us towards the right scaffolds, either natural or synthetic. Those are both appropriate, but both spaces are dry at the moment, mostly dry.
Can't lie, I'm really loving this overlap of the term brute force between the cyber realm and the biological realm.
Yeah, I
Yeah, I'm
guess.
just using it because I think Germany was very mad that they spent a lot of money and a lot of manpower, and then the person that discovered the really effective one was
It's
it was
appropriate.
very
It's appropriate. I mean, we're living in a realm, we're living in a time of merged biology and technology. So, yeah, brute force it. Yeah, I love it.
Yeah, I compare a little bit, the machine learning approach with back in time, what the Germans did at those companies with what, uh, I compare a little bit that with that, just, not thinking, just screen whatever you have and throw it on there. I mean, yeah, it's maybe both work, but not every country can afford that type of brute force approach. I think it's not realistic.
Plus, recently, it just leads to the discovery of the same antibiotics we already know about.
Yeah.
So we have a huge problem on that area. You spend all this money, you do this brute force approach, you spend time to screen, whatever, and then you come back to, oh, well, we know about this already. So yeah, we need to do something outside that realm, I think.
Yeah. Again, thank you so much. I'm not sure if anyone has another question. Please go ahead.
Sure.
We have been going for an hour and 15 minutes. Thank you so much for being so generous with
It's
your
been a pleasure.
time today and for coming. I know, joining Clubhouse and everything is really, thank you.
That's my fault for being adhering to old technology, but thankfully my husband's phone works. So
No, thank you for going through all the trouble for
sure,
coming.
no problem.
Really appreciate it.
Yeah, no, thank you. Made this Friday extra special, knowing that there is some salvation against all the threats from the microscopic world. So, a little bit of hope on a Friday. And now we can all go celebrate or relax or whatnot. So, thank you for joining us.
Absolutely, it's been a pleasure.
Thank you.
Thanks, everybody. Have a good
Thank
weekend.
you. Have a good weekend. And thanks everyone for coming and asking great questions. And please join if you like rooms like this. Please join Science Society. And we will have more rooms with great guest speakers, with real scientists that do real research. That you can interact with. We have actually tomorrow, let me just tell you, we have tomorrow 2 rooms. One at 12 p.m. EST, Dr. Pridarshini, who had a recent publication about suppressing genes across generations. It's a Nature Methods paper, so she will be presenting. And then at 9 p.m. tomorrow, you have Dr. Lee from NIH with his recent publication where he shows that mitochondria act as little microlenses in cone cells, which I think was really interesting. So, and he gladly agreed to come. So, yeah, and then on Monday we have 2 scientists that will explain to us how spiders fly. It will be really interesting. Um, that paper, and then how neurons find their place. And then on Thursday we have a really great addition that came in today with Dr. Liu. he developed a new theory of entropy solving material designs that will enable us to produce way smaller new gadgets. And so yeah, I'm really glad he agreed today to come. I met with him, so it will be another exciting week, and we are not done yet. Tomorrow will be 2 interesting rooms, so join us again, and thank you everyone.
We'll just have to hang in there for the spider information. Haha.
Oh yeah, that paper is so cool. Let me just go. Yeah, I'll on the Twitter announcement I'll share the paper. It's really cool. So yeah, I'm happy they agreed to come. So they joined the club, and yeah, so I could add them to the room. So I'm really looking forward to having them. Okay, enjoy the rest of your Friday wherever you are, and hear you back soon. Thank you, everyone.
Awesome. Take care, everybody.
Bye.
Thanks, Katerina. Do follow the Science Society. There are so many interesting talks coming up.
Yep, exactly. Thank you so much, Frank. Thanks for helping everyone. Bye.